Okay, now recording started. No issue. Now the next question is similar to that.
Sir,
Yes.
I have one doubt about you told that trypsin cleaves at basic amino acids, but
Yes.
you only determine lysine and arginine. So what about histidine?
Histidine normally, why it is not cutting at the histidine side? Because histidine is not always acidic. That is the general nature of histidine, right? It can act as a both acid and base. Because its pK value lies in the range of physiological pH that we discussed in the great detail in earlier lectures. And because of that only you will find histidine in the most of the enzyme binding site, like the active site of most of the enzyme. Because if it is surrounded by the negatively charged amino acid residue, means if it is surrounded by the acidic amino acid residues, then the histidine will occur as a positive charge because it tries to neutralize those charges. And if histidine is surrounded by the positively charged amino acid residue, then it will act as a base. Okay, so because histidine is not classified in either base or acid category, because it can act, it switches its characteristic based on the surrounding. So due to that, the trypsin in the trypsin cleavage site, histidine does not come in play. Is it clear? Because
Yes,
of
sir.
this property only. Because of this property only. So now the next question here, based on the previous discussion, you have a trypsin digestion here. You have a trypsin digestion. Now tell me which, how many peptide sequence you will get if you digest this peptide bond, this peptide sequence with trypsin, then how many fragments you will get? 4, 2, 3 or 5? How many?
Two.
Where will 2? Why? How many amino acids are there? Basic amino acid residues are there?
Lysine and arginine. K for lysine, R for arginine.
Okay, so which fragment will you get if you digest this? So which fragment will you get? And can you name me? Where will
First
it
one,
cleave?
M,
Okay,
V.
M,
It leaves
V.
after K. So M, V, D, Y, T, G, K.
So K will come with this fragment or K will go with that fragment? Where it will cleave? Here or here? Where will it cleave? Here or here?
After K.
After K. Why? Why? Why after
Trypsin
K?
cleaves. Trypsin cleaves at after C terminus. So
At the C terminus. Yes. So if protein is written here, so we already know that left hand side you have which terminal, C terminal or N terminal?
N terminus.
N terminals. Yes. So in the left hand side you will have N terminal, and the right hand side you will have C terminus. Okay. So basically the C terminal will be on this side. Like the lysine C terminus will be on the right hand side. So this will cleave at this side. So K will come here. Then you have T, M, S, L, R. So will it cleave here? Because arginine is also there. It will not cleave here. So you will have two fragments. One and this two complete. You will not have T, M, S, L and then R. Because on the right hand side, there is on the C terminus, there is no other amino acid available. So the two fragment will be MVDYDGK and TMSLR. Correct answer is 2. Very good. Now the next question is what is the active site of chymotrypsin? Because in the while ma'am was discussing the chymotrypsin mechanism, so there she discussed about it. It has catalytic triad. It is a family of serine protease, right? It is a family of serine protease, and in its catalytic triad, how many amino acid? Which will be the 3 conserved amino acid residue here in
Option
catalytic
A,
triad? Option
histidine,
A,
aspartic acid, and serine.
and that will be the place histidine at the place of 57, aspartic acid at the place of 102, and serine at the place of 195. Very good. Now, in a compound, you have NAG and NAM of the peptidoglycan. Then which enzyme will you use to cleave this? That
Lysozyme.
lysozyme. Very good. Now the next here is the complete set of enzymatic cleavage site. So here there are various peptide proteases. Aminopeptidase is another family where in the why the cleavage is always called hydrolysis reaction? Why it is hydrolysis? Why protease cleavage are hydrolysis reaction? Can anyone tell? Anyone?
Peptide proteases cleave peptide bonds. So our peptide bond is cleaved by addition of water molecule H2O. So
Yes. In the next cycle, like when the serine act on the its cleavage site, and then in the next cycle when the acyl intermediate have been formed, tetrahedral intermediate, there is a requirement of water, which in which the histidine will abstract the proton, and then hydroxyl ion will be created. So because of that, because of the role of water is there in the catalytic site, it is this protein cleavage, peptides cleavage are known are also called as the hydrolysis reaction. So aminopeptidase A, the cleavage site is it releases the N-terminal leucine, but it may also be other acids including proline, but not arginine and lysine. Arginine and lysines are cleaved by the trypsin, right? Aminopeptidase B, it releases the N-terminal arginine and lysine from the oligopeptides when P1 dash is not proline. What does it mean? Releasing of N-terminal arginine and lysine means it will cleave at the C terminal. Okay, it will cleave at the C terminal and it will release N-terminal of arginine and lysine. So that means the aminopeptidase B is also a class of trypsins. It acts as a trypsin kind of activity. Aminopeptidase B. Now coming to carboxypeptidase A. Carboxypeptidase A releases C-terminal amino acid, but little or no action with the aspartic acid, glutamic acid, arginine, lysine, or proline. Okay, that means it is cleaving at the N-terminal. Only carboxy in the enzyme family, when you see the carboxypeptidase cleaves at the N-terminal and releases C-terminal. However, other enzymes cleave at the C terminal and releases N terminal. Okay, so chymotrypsin preferentially releases N terminal of tyrosine, tryptophan, phenylalanine, leucine at the P1 position. Papain preference for an amino acid bearing a large hydrophobic side chain at the P2 position. Now pepsin cuts the preferentially at the hydrophobic and preferably aromatic amino acid. Thermolysin releases C terminal. That means it will cut at the N terminal of leucine, phenylalanine at the P1 position, and trypsin preferentially releases N terminal, cleaves at the C terminal of arginine lysine, but except when the proline is next to the any of these basic amino acid residue. So the next question is, below mentioned enzyme belongs to a which class of family? Is it it is converting malate to fumarate. So which class of family it will belong? Oxidoreductase, transferase, isomerase, or lyase? It is doing the isomerization reaction, right? Both are the isomers, malate and fumarate. It is not this particular enzyme is not adding any group or any atom, or it is not cleaving anything, but it is transforming one form to another form. So it is called as a this class of enzyme will be isomerase, right? And what you will call it? You will call it malate isomerase or fumarate isomerase based on the time where it is converting. Okay. Next question is, in deoxyhemoglobin HB, the ferrous ion, which is Fe2 plus, is 5 coordinated to where? चला गया क्या? पर इनका रोज का नाटक हो गया है. Yeah, there was some internet connectivity issue. So I was talking about this question. Question number 8. In deoxyhemoglobin, in the deoxy state, the ferrous 2 ion will be coordinated to where? 4 nitrogen of heme group to an oxygen, or 2 nitrogens of heme and 3 histidine residue in HB, 4 nitrogen of heme to a water molecule, or 4 nitrogen of heme to the proximal histidine of hemoglobin. Which is correct? In deoxy form of hemoglobin, where the ferrous ion is coordinated? The correct answer is here. 4 nitrogen of heme. Heme is 4-pyrene ring, and then the proximal histidine of HB. Okay, in the deoxy form, when oxygen is not bound, the ferrous 2 ion will be coordinated to the nitrogen group of the heme, and then proximal histidine of the hemoglobin. Okay. Now coming to the kinetics of hemoglobin and myoglobin binding. You have studied that hemoglobin and myoglobin. Okay, so but the difference is that hemoglobin has four hemoglobin has two identical polypeptide chain. However, in myoglobin you have only beta type chain of hemoglobin. Okay. However, in hemoglobin, you have four different subunits of this beta type. Okay, so it is a tetrameric protein, hemoglobin. However, myoglobin is a monomeric protein. If it is monomeric protein, then it can only bind to the one oxygen molecule. However, hemoglobin has four subunits, so it can bind four oxygen molecule. Okay, so this is the difference between hemoglobin and myoglobin. Now, hemoglobin and myoglobin, both their characteristic depends on the binding characteristic. How, like where it will release, where it will be bound with oxygen, that totally depends on the its binding characteristic. Now, you have already studied that hemoglobin is hemoglobin shows cooperativity of positive cooperativity, right? So here is a graph given. Now in this graph, when you see a positive cooperativity means that if one particular oxygen molecule is binding to one chain of hemoglobin out of four, it will facilitate the other oxygen molecule to bind to other subunit, and it will rapidly bind to their phase. Okay, why it is essential? Can somebody explain? Why oxygen? Why when one oxygen binds to the like hemoglobin, one subunit of hemoglobin, then it facilitates the oxygen binding with other three subunits. Why it is beneficial in physiological case, in biological conditions? Can anyone explain me that? That is required. That is required because when there is a low concentration of oxygen present in the environment, which is in muscle. In muscles, normally partial pressure of oxygen is very low. Okay. However, in case of lungs, there are lot more oxygens present. Okay. So what you will see that myoglobin has only one binding affinity, and its binding characteristic is hyperbolic. What do you mean by hyperbolic? Let me go through this slide. Yeah. So here the two structure of myoglobin and hemoglobin is given. Now, in case of when the oxygen partial pressure, okay, here in the y-axis you will see the oxygen saturation. Oxygen saturation means when you treat this as a protein ligand interaction. In protein ligand interaction, your protein is myoglobin. When your protein is myoglobin, when the protein is myoglobin, then what you will see, myoglobin will bind to only one oxygen molecule, right? So that means during the binding, when myoglobin is there, it binds to oxygen and it will follow this MO2 characteristic. Now, when you write the KD of this, the KD means dissociation constant. Dissociation, why we are writing dissociation constant? Because the dissociation constant will tell you at which place at which condition the myoglobin will release its oxygen. So the Kd will be, in case of dissociation, Kd of myoglobin will be partial pressure of oxygen, then myoglobin concentration divided by bound myoglobin, right? Okay, so the oxygen saturation here shows that where the binding, when the binding is happening, when binding is there, like, and in the blood, how much oxygen is available. That you, so because oxygen is a gaseous molecule, so the expression of concentration will be partial pressure of oxygen, right? Partial pressure of oxygen in blood. And what you see that myoglobin follows a hyperbolic characteristic. Hyperbolic characteristic means its Kd value, means Kd, when there is a like the, it will be, myoglobin will be saturated even at a lower concentration. That means in the muscle condition, when in muscles, normally in muscles, the partial pressure of oxygen is very low. Okay. In muscles, the partial pressure of oxygen is very low. This is muscle. And here you will see lungs. These are lungs. In lungs, your partial pressure of oxygen is very high. The oxygen concentration is very high. However, in muscles, you have very low concentration of oxygen. So in that case, what you will see that myoglobin is still, even at the lower concentration of oxygen, the myoglobin is 80% saturated. This is fractional saturation. That means one denotes 100%. Okay, so even at the lower concentration, means lesser than the 10 parcel, like 10 millimolar per Hg. Okay, 10 torr, even at the lower than 10 torr, 80% of the myoglobins are saturated. However, in case of hemoglobin, this is not the case. Hemoglobin will bind slowly. It slowly. How? Like in the muscle case, it need, if it is if oxygen is bound to the hemoglobin, that means it will not facilitate the metabolism of, and it will not generate energy. Okay, because oxygen are the terminal electron acceptor in electron transport chain, right? So if oxygen is bound to hemoglobin, then the respiration will not occur and cells will not get energy. Okay, respiration. What does the respiration mean? Respiration mean in the electron transport chain, when oxygen accept accept the electron and finally water is being converted and you get ATP in mitochondrial matrix. Okay. When you get ATP, that is called the complete respiration. Okay, complete. So what you need to understand that when you need energy, then oxygen need to be in free form. It should not be in bound form. Okay, that is what hemoglobin does. At the lower concentration, hemoglobin does not, like oxygen does not bind to the hemoglobin. It will be in the free state. However, when the oxygen concentration is very much high, then which is in lungs, means at in the lungs, the hemoglobin will be like all the four sides of hemoglobin will be occupied, means the hemoglobin will be totally saturated. And while transporting means when a hemoglobin takes up all the oxygen. It occupies the all binding site. All binding site of hemoglobin is occupied by the oxygen at its at it when the oxygen saturation is when the oxygen concentration is very high in the lungs, it will be bound, and when it goes to the muscles, it will release all its oxygen content. Means from lungs to muscles, it is transporting. That is a good characteristic of a transporter. However, in case of myoglobin, it is not a transporter. It actually stores the oxygen, because and myoglobins are present in muscles. Myoglobin will only lose when the partial pressure of oxygen is very low. That means when does it occur? When you are climbing, or you are doing, when you are running, and then when you are running to your destination, and then you are panting. At that time, myoglobin will release its oxygen, because at that time your muscles are working very hard. Okay, when your muscles are working very hard, they need more and more oxygen. So hemoglobin will not be able to sufficiently provide oxygen in that condition. So when the oxygen pressure is very low, in that condition, myoglobin will release its oxygen, and then your cells will be able to metabolize your and it will be able to provide you energy. Okay, is it clear? So which is the correct state you need? Oxygen bounded state, or oxygen released state? Which is beneficial to you? Bound oxygen state, oxygen bound, or oxygen free? Oxygen is in free condition. Which condition you will get more energy? The question from this discussion is that. Can anyone answer this one? When you need more energy, what do you expect? Oxygen should be bound to the hemoglobin, or it should be in free form when you need more energy? Anyone? Sakshi, Pragna, Zaid, which will be the beneficial condition? When you need more energy, in that case means your metabolism need to be fast and you need to generate more and more energy. So in that case, oxygen need to be in free form. Okay. And when there is lot more oxygen is there, then this oxygen need to be transported from one place to another place, and that role is played by the hemoglobin. Hemoglobin will bind and it will release it oxygen when there is a low oxygen concentration, such as in muscles. Okay, so and this oxygen and myoglobin will only release its myoglobin will only release its oxygen when the partial pressure of oxygen is very low, because its KD value, if you see, is very much low. Means even at the very lower concentration, even very lower concentration of oxygen, it is 50% saturated. Okay, that means myoglobin will bind the oxygen more strongly. It has the higher binding affinity. It has the higher binding affinity and lower dissociation constant. Lower the KD means higher binding affinity. Okay, so here this percentage saturation is given here. This is similar to this. Percentage saturation means here at the 100%, like in case of this graph is for the hemoglobin. So when 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 there is a right shift of the hemoglobin curve, it means the affinity will reduce. However, when there is a lower temperature means when you go to a colder environment and you have higher pH or lower CO2, in that case hemoglobin will be strongly bound to the oxygen. Okay, higher pH case means you have ample amount of H plus ions in your in your ample amount. Higher pH means less H plus, right? In case of higher pH, you have less H plus ion. Lower CO2 means your body is not able to metabolize very fast. Okay, so lower CO2 means your oxygen concentration will be very high. So in that case, your binding affinity of hemoglobin will be increased. So that in that case, the curve will be left shifted. However, when you have higher CO2 means you have less oxygen. You have less oxygen means your body is producing, body is metabolizing more like in more rapid rate, and at the higher temperature case your oxygen concentration will be lower. In that case your uh, hemoglobin binding affinity will be reduced and hemoglobin will drop off its oxygen. Okay. So that your body can generate more and more energy. That is why when the temperature is warmer, in that case you sweat more. Okay, and you try to get more and more oxygen, because at higher temperature the oxygen concentration in your body will be lower and you will be feeling not easy while metabolizing. Okay, is it clear?
Sir,
Yes.
which one has high affinity for oxygen, myoglobin or hemoglobin?
Higher affinity for oxygen is myoglobin. Myoglobin is strong. This is this is what is given here in this graph, right? In this graph, the how do you define binding affinity? Binding affinity is K inverse of KD, dissociation constant, right? Binding affinity. If you talk about the binding affinity, KB means it will be having the lower KD. It is inverse of KD, dissociation constant. Now, what is dissociation constant? Dissociation constant means at which at the concentration of oxygen at which the 50% of oxygen is detached from this protein. Okay, so for myoglobin, the half of saturation is here. Half saturation is here. However, for hemoglobin, the half saturation is somewhere here. It means the KD value, the hemoglobin will have the KD value of around 40 torr as per this graph. However, the KD value for myoglobin is about 5 torr. The lower KD value means higher binding affinity. That means myoglobin has higher binding affinity to the oxygen, and hemoglobin has lower binding affinity to oxygen because its KD value is very much high. Higher KD means lower KB, lower KD, higher KB. Is it clear?
Bu- But sir, in assignment, they give an option. Hemoglobin has higher affinity for oxygen than myoglobin, and it
That is
indicates
wrong.
correct option.
That is wrong. That will be corrected. That is wrong. I will also discuss those problems. So after this all concept clearance, I will go to the assignment problem, and then we will see if there are any clarification needed. And then I will also email to the other TAs of this course so that they can recorrect the questions if you have lost any mark due to the wrong answer. Okay.
Okay, sir.
Yeah. So here it is given the oxyhemoistate and the deoxyhemoistate. So these are the heme prosthetic group. So this is the heme for firing ring. So this in this porphyrin ring, like there are four rings, this ring, and then these four nitrogens are coordinated with the ferrous Fe2 plus ion, right? And this is supported by histidine residue. Okay, so in the deoxy form, it is not bound. However, in the oxy form, this ferrous ion will be coordinating with the oxygen binding. Okay, so this is in the depiction form. For this is the deoxyheme and this is oxyheme. Okay. Now coming to the previously, yeah. This we were talking about the cooperativity. In the case of cooperativity, hemoglobin sense a positive cooperativity. That means initially when oxygen is not bound at this stage, till this stage when oxygen is not bound, they like they will it will be slower kinetics. Okay, it will be having the slower kinetics, but when one oxygen is bound, it will take a sudden leap, and you will have a positive cooperativity here. Means more and more oxygen will bind, and then again when all the active site is when all the binding site is occupied, then again this will slow down. So it has three phases. One where it has zero cooperativity, then it is third when again it has zero cooperativity, but in between it has cooperativity of n in between 1 to 3. Okay, it is approximately 3 for hemoglobin. The cooperativity is approximately 3. Okay, so this is the kinetic equation given here for where n denotes the number of binding site in protein. Okay, because the binding site in hemoglobin is totally 4 binding site, right? So the equation is derived such as here. The Kd is equal to Kd will be P L and L to it will be N. So when you are writing a dissociation constant, equilibrium constant, so it will be concentration of protein. For dissociation constant, it will this will be the product and this will be the reactant, right? So the Kd will be concentration of protein, then concentration of ligands means oxygen to the power n divided by bound form of oxygen, bound form of protein. Now, when you are writing the partial saturation, partial dissociation, which is Y, that will be ligand to the power n divided by ligand to the power n plus Kd. Okay, this is known as the Hill equation. Okay, Hill equation is giving you the partial saturation, the equation between the partial saturation, and when you take the log of this, okay, when you take the log on both of side, then you will get the log of theta divided by 1 minus theta, where theta is the partial saturation, and then you will see this n log this n log L minus n log n. Okay. And this is in the form of y is equal to mx plus b. So on the y-axis you have n log this is your y, y is equal to mx plus b. Okay, so y will be this. Y is your log of theta divided by 1 minus theta, which is partial saturation, log of partial saturation. Then on the y-axis, you have partial pressure of oxygen. Okay, so for the cooperativity, which is N or H, when ma'am was talking about, so for Hill coefficient, she said N H. So here, in case of hemoglobin, what you will see at the initial condition, your binding is not much efficient. When no other oxygen is bound to the hemoglobin, then your Hill coefficient will be 1. Okay, but and that will be the case of when it is completely saturated also. When it is completely saturated, then again the Hill coefficient will become 1. Means then it will not show the cooperativity. Here also it will not show the cooperativity, but in between, when when one oxygen has bound to the one subunit, then it will show the cooperativity and rapidly it will adopt complete oxygen binding. Why? Why it is important? Because when uh, as you know that oxygen is only bound to the hemoglobin in the higher oxygen when there is a higher oxygen concentration present in the environment. Okay, so when one oxygen is bound to it, then it will facilitate. That means now the hemoglobin is sensing that there are lot of oxygen concentration available. So it need to store and transport it from one place to another place. So in that state, it it transition from the T state to R state, and this phenomena is very rapid. Okay, and during this period, you will have slope of n is equal to 3. That means the cooperativity is around 3. Okay. Now, coming to this question for hemoglobin, the question is regarding myoglobin. You have partial pressure of oxygen is 10 times of partial pressure of P50. P50 is the concentration when 50% of the myoglobin is saturated. So partial pressure of oxygen is 10 times of partial pressure of oxygen. Then what will be the fractional saturation? Can someone calculate it immediately? Uh, what will be the fractional saturation in this case? What is the equation of fractional saturation? P bound to the oxygen, it will generate PO2. Okay, so fractional saturation will be YO2 is equal to YO2 will be this is your homework. Okay, you calculate it. This is your homework, how to solve this problem. This is your homework, and tell me the equation of fractional saturation. Okay. Now coming to the next question. This is regarding to the hemoglobin. So the dash histidine is attached to the ferrous ion in hemoglobin, is connected to dash helix, while dash histidine is connected to the dash helix. There are different. If you have studied the structure of hemoglobin, then in that you will have a like two different like four different side chains, four different side subunits are there in hemoglobin, right? So if you are focusing on one subunit and it has a heme group, so in that heme group, in that one subunit, it will be having the different foils helix. So these are classified as the helix. Okay. So normally, if you study this question, like if you study the structure of hemoglobin, then you will see that the histidine, proximal histidine is attached to the ferrous ion. So here in this where was that? Where I showed you. So here in this prosthetic group, this histidine is proximal histidine, and this proximal histidine is attached to the ferrous ion, right? Proximal histidine is attached to the ferrous ion, and this is attached to the F helix, while the distal histidine, which is distant, that is connected to the E helix. So option A is correct here. Now this we have discussed. Now we will discuss the role of BPG. BPG is bisphosphoglycerate. What does the bisphosphoglycerate does? It regulates the binding of oxygen to hemoglobin, right? How does it do? Can you recall? Can anyone recall how BPG regulates binding of oxygen to the hemoglobin? Bisphosphoglycerate are generated by the red blood cells. Okay. Normally, cells do not want to reduce the number of hemoglobin. Okay. Cells do not want to reduce the number of hemoglobins in the body, red blood cells, because hemoglobins are found on the surface of red blood cells, right? Because of presence of hemoglobin only, red blood cells looks red. Okay, because it has ferrous ion and 4 ring. This heme group gives the red color to RBCs. Okay, so let us say when you have a condition when you are climbing a mountain. So when you are climbing a mountain, at that condition, you will at the mountain when on the peak, the concentration of oxygen is lower or higher.
Lower.
Lower. Yeah. So when in the surrounding, the oxygen concentration is lower. So in that case, what do you suggest? A hemoglobin will bind to the oxygen or it will not bind to the oxygen?
Not bind to oxygen.
Yeah, because body needs oxygen for respiration, right? For metabolizing and for generating the energy, ATP, right? So, uh, so what? So when you study a protein ligand chemistry, there you will see if you want to reduce the concentration of protein ligand bound states, then what you can do, either you can reduce the number of proteins or you can reduce the number of ligands. Okay, but ligand is already lower in case of when you are above the cliff, then ligand means oxygen is already lower. Then you want to shift this phase to lower hand side. Then will you what will you suggest? Will you lower down the concentration of hemoglobin? Because hemoglobin will be required. These are the proteins. These will be required in the later stage. So what body does in stage of reducing the number of hemoglobins, what they do, the red blood cell itself start producing BPG, which is bisphosphoglycerate. Now this BPG has the higher binding ability than oxygen to the heme group, and this BPG go and bind to the hemoglobin. so that the oxygen cannot bind to it, and oxygen is available in our body, and our our body can do the respiration normally. Okay, so here is the case when BPG is lower, then in that case the KD value is lower. That means oxygen is strongly bound. But when you are increasing the concentration of BPG, when you are increasing the concentration of BPG, you see the increase in KD value. That means the the hemoglobin will release oxygen rapidly. Okay, means it will not bind to the oxygen, and oxygen will be available in the cell. Now you have increased more BPG, then again the KD will increase. So KD will increase means the binding affinity of oxygen to hemoglobin will reduce. So this is adopted when you are at the higher altitude. At the higher altitude, the BPG the blood will produce more and more BPGs, so that the hemoglobin is not able to hold the oxygen molecule in bound state, and oxygen will be in the free state of the cells. The similar case you will see in the case of venous blood and arterial blood. Venous blood here talks about the blood in vein, in that which carries like red blood cells, more RBC, which is the pure blood, arterial or venous? Arteries carries the pure blood, or vein carries the pure blood? Which one carries the pure blood? Anyone? If you have studied the physiology, human physiology, so in human physiology we will see that arteries are that who carries the pure blood. Pure blood means the oxygen bound blood. So in arteries the oxygen concentration is very high, so in that case hemoglobin will be completely saturated. And in venous, which carries more of carbon dioxide, so in that case the uh, partial pressure of oxygen is lower. Okay, and your hemoglobin will act as a carrier. Okay, so the same thing is denoted here. In the venous blood, the oxygen concentration is lower. However, in case of arterial blood, the oxygen concentration is higher. So hemoglobin will be in the bound state. However, in the case of venous, the oxygen will be in the free state. Now, this is the equation for binding kinetic of oxygen to myoglobin. Okay, so this is the KD, myoglobin, partial pressure of oxygen divided by the bound state, and fractional saturation is this one. So the fractional saturation will be bound state divided by myoglobin concentration plus KD value. Okay, so Y will be partial pressure of oxygen divided by KD plus partial pressure of oxygen. Now, using this equation, you can solve that question which I had given earlier. Okay, so the amount of O2 bound to myoglobin is function of both oxygen concentration and affinity of oxygen to the oxygen myoglobin. Okay, so in that question, what was given? The partial pressure of oxygen is 10 times of P50, which is K. Okay. So what will be your fractional saturation? Your fractional saturation will be Y is equal to PO2 is given 10 times of P50 divided by K plus 10 times of P50. So your fractional saturation will be how much? It will come around 0.9. So the correct answer was 0.9. Okay. Now you based on this equation, you solve that problem and your answer is 0.9. Now let me go to the like understanding how you do this protein ligand docking. Okay, and how do you understand this whether the protein is bound to the ligand or not, how you understand the interaction between the different amino acid residue with the ligand, and how do you visualize it, what all references you can get. So normally to study the autodock, to study docking means to study the protein ligand interaction. To study the protein ligand interaction in silico, you need a software which is Autodock tool. This is a very much diversified tool which is used in most of the computational biological like most of the computational biologists use Autodock tool for docking because uh, in this you can set parameters according to you. Then for visualization you can use PyMol, and you can also use Sys PDB viewer. Leak plots, this software you use when you are studying, when you are understanding that if you have performed the docking, then which all amino acids are interacting with your ligand molecule, that you can visualize using like leak plot plus, and that you will get in the 2D plot, 2D representation. Okay, I will show you this all a kind of a demo. Then another software which you can use, that is protein ligand interaction profiler. Philip, protein ligand interaction profiler. In this, you can this will profile all kind of like hydrogen bonding distance. Then which particular uh, ligand the atom is binding with which particular amino acid atom. Then P2 rank is used for identifying the binding pocket in a particular protein chain. Okay, so the docking protocol is how you perform a protein ligand docking or enzyme substrate docking. How do you do that? For that, first you need to procure the PDB. PDB is protein data bank file. Protein data bank file of your protein. Then you have to procure the ligand molecule. Now you perform the docking using Autodock tools, and then the Autodock will result a DLG file, .dlg file, and you analyze that DLG file. You convert that into the PDB format, and then you subject that PDB file to any software, visualizing software such as Philip or Lickport Plus or PyMol, and then you will see the binding, how this binding is happening. Okay, so to give you a brief how one can follow these all processes, I will take example of a molecule which is let us say chymotrypsin. I am taking. How you will do? You first go to the protein data bank. I will show you the demo. You go to the protein data bank. Protein data bank, PDB data bank is the home where you can get the crystal structure of any of the protein which have been solved. Okay. using X-ray crystallography or NMR spectroscopy, solution NMR. So let me search chymotrypsin here. So chymotrypsin C, any structure. So there are two structures of chymotrypsin C available. You can download any one. This is the ternary complex of protocarboxypeptidase, proteinase, and chymotrypsinogen. Okay, let us visualize this structure. Is it recording? Yes, recording is going on. Okay. So once you open this one, then go to the visualization. Structure visualization. Visualization you can do in the PDB also, and then you can do in the PyMol and other visualization software also. So here it is represent, this chymotrypsin is represented in beta sheets, random coils, and alpha helix. This is what alpha helix look like in 3D. This is your alpha helix. This is your random coil. This is your random coil, this coils structure, and your beta sheets are here. These are the beta sheets. Okay, beta strands. Now these dot molecules are nothing but crystal water. Okay, and let us see which molecule it has bound. This is a phosphate ion. Okay, you will see in the like left hand corner here. Here you are in the right hand corner. You will see this is a phosphate ion. Now when you rotate it, there are two phosphate ion bound to it, right? Two or three. Now if you want to visual, and the amino acid sequence is given here. When you hover here, like any of this, the particular chain will pop up. Okay. If you are giving here, what is this? What is this fragment? If you select it here, so this is your leucine 27. Okay. Now, if you want to visualize and perform the docking, now if you want to visualize and perform the docking in that, let us say, let us see what kind of interaction it is having. Okay. So here, let me zoom. Let me remove the crystal water. Then ions also, ions. There are three ions, phosphate ion. Okay. And now see this kind of interaction is there. These are the side chain of leucine E C, and this is tyrosine. Tyrosine 29. This is your histidine 28. Okay, this is histidine ring. Now this is again your tyrosine, right? Tyrosine 24. Here you have glutamic acid 23. Okay, now see how this is coordinated. See how what is the distance? This is the distance between phenylalanine 25 and phenylalanine 28. So this is the intermolecular distance. Now this distance is for the tyrosine, this tyrosine 29 and phenylalanine 10. So this is how you see the interaction. Now let us perform a molecular docking. How you will perform a docking? So for that, what you do, you download this structure in PDF format, download file of PDF format. Here are the different sequences, FASTA sequences there. But for the crystal 3D structure, you need to download the PDF format and save it somewhere. This is 4H4F. 4H4F is the PDV ID of crystal structure of human chymotrypsin C. Okay. Now this has been downloaded. The PDF format is downloaded. Now let me open PyMol in my computer. All the softwares that I will use here, all the softwares are open source. That means if you download and install, you won't need any, you won't have to pay any money for it. Okay. So now open that structure that I had saved here, desktop demo. Yeah. See, this is how the chymotrypsin, human chymotrypsin look like. It has 3 phosphate ion bound. Now, if you want to visualize the sequence, you visualize the sequence here. So it has how many amino acids? It has 2 chain, right? It has 241, 242, 243, 244. In one chain, it has 244 amino acid, and another chain also it has how many amino acid? For the chain B, it has 66, 67, 68, 69, 70. It has 70 amino acid residue in another chain. And then it has another C chain. So it has total how many chain? Chain A, chain B, chain C. Chain C is the smaller one, and it has one total 6, 7, 8, 9, 10. So it has 10 amino acids in chain C, then 70 amino acids in chain B, and in chain A it has how many amino acids? Total 244 amino acids. Okay. If you select any of these, and you can in PyMol you can do any you can do any kind of visualization. Like these tabs are for visualizing any state. Like here, if you want to show all complete structure as a surface, you click as a surface and it will appear as a surface. Now in this surface form, you can see the binding pockets. Okay. Now, do you remember what was the binding active site of chymotrypsin? What was the binding catalytic site of chymotrypsin? It is a serine protease, right? Can you recall? Anyone? Sakshi?
Yes, sir.
So what was the binding active site? I will mark that and then you can visualize it where it occurs. Okay. Which position of serine is the active site of chymotrypsin?
160.
100. Serine 115, right? Is this one or which one? In the question also I had given, right? For chymotrypsin, this is the chymotrypsin only, right? This we downloaded the chymotrypsin
195.
structure, right? So it was 195, serine 195, aspartic acid 102, and histidine 57. Okay, let us mark that here. So 195 on the 190, this is 191, 192, 193, 194, 195. On serine, it is 195. So here, let me show this as a hide surface. Okay, so and then I will mark this as a, and the another one was serine 195, then histidine, which histidine? Histidine
57.
57. So go to the position of histidine 57. This is 56 and 57. 57 you have H histidine. So see, both are in the proximity, right here. Now another one was aspartic acid. Which one? Aspartic acid
102.
102. Let us see in aspartic acid we have or not. 101 you have N is for N is for what is the N?
Glutamine.
N denote glutamine. Glutamine is N or asparagine.
Asparagine. Sorry.
Yeah. Glutamine is Q. Asparagine in N. And aspartic acid is, what is the one letter code for aspartic acid?
D.
D. So at 102 position, you see a 1D also here. So this is what ma'am was talking about. So all in this chymotrypsin structure, all three are here. And now whatever we have selected, we will rename it as an active site. Okay. Rename as active site, and then we will change its representation so that you will be able to visualize more accurately. Now we will show this as a cartoon, not cartoon, sticks. See, and we will like so the we will hide this structure and we will show this one sticks. See, so these are the three amino acids that play an active role in the active site of your chymotrypsin. And if you want to know what are the poison here, you can label also. See, residue 1 letter code. Are you able to see it? See, this is serine 105, serine 195, aspartic acid 102, histidine 57. So this is your binding site. Now what we will do, now we will show complete structure as cartoon, and then we will see where it fits. And then we will show this as a binding pocket. Okay, surface. We'll, we'll come. This as a surface. See. So here your hydrophobic pocket is there. And then here the main chain will lie. Right. And then serine will do the cleavage. Let me yeah. So here. So can you see this groove? This kind of groove? Are you able to see this? So here your peptide chain will come. If you dock with this a peptide chain, then your peptide chain will come here. Your hydrophobic pocket like phenylalanine, tyrosine, and tryptophan, they will bind here, and then serine will cleave at this site, and your product will be released. One your peptide chain will be cleaved in two fragments. So this is your kind of binding site. And active site residues are in this pocket. Is it clear?
Yes, sir.
Yeah. So this is how you do the like this is the visualization. Now, if you want to check the binding pocket, if let us say you don't know the binding pocket of any protein, then how you can check that? You can check that in the P2 rank that I have given, right? In the slide I have. So go to the Prankv website, put the PDV code of that protein. PDV code of this chymotrypsin was 4H4F, right? Put 4H4F and then use original structure. Now see, it will show you the different binding pockets, and based on the score, the highest binding score, we will be able to identify which is the correct binding pocket. So it has suggested that is it how many pockets are there? There are total 3 binding pockets. Okay, and these are colored. So the highest score of binding pocket is this one, right? 9.43. Another binding pocket is 6.29, but this is pocket 3 is very lower. So that means these two are the most probable binding pockets because its pocket score is very high, 9.43. So we will uh, like hide these two. Okay, and then we will only see this one. So this red one is the binding pocket. So, and what are the amino acid involved in this binding pocket? That also you can see here. See, you will see the involvement of this is your pockets. This is your pocket one. Pocket one is gray color. See, so it has serine 195 here, right? Then in the binding site itself, you will find one more. Let me zoom. Which is this amino acid? This is crystal water. This amino acid is arginine 53. So in this binding pocket, there are total 17 amino acid residue. But the catalytic residues are serine 105, serine 195, aspartic acid 102, and histidine 57. Rest all are there to stabilizing, yeah, to making the hydrophobic bond or hydrophilic bond to like to make the protein chain in its place. Okay, so this is how you visualize the binding pockets. Now, once you have decided, once you have like understood the binding pocket, you can download also. You can download the data, complete data. Then, if you open it, predictions. So it has three binding pocket, and their amino acid residue also has given. Like in binding pocket 1, 147 is there, 148 is there, 192, 216, 217, 217A, then 219, 220, 43, 44, 45, 53, 55, 66, 68, 70, 69, and 70. These are the amino acid residue which are helping the helping the different your ligand molecule to stay in place. Okay. So once you get this binding pocket data, this around this binding pocket data, you will create a grid box and then you will do a docking. So what is the docking protocol? I will show you a docking protocol here. These things I have shared with all of you. If you like move to the folder, then you will able to see the docking protocol. How do you do the docking? So for docking, you will need a protein structure. Then what you do, you open that in the Swiss PDB viewer, and first thing is to minimize the energy. Okay, first thing, you need to minimize the energy of your protein molecule. Then how you do that? You do by select all and then control N. That is the control N shortcut is to rearrange the complete amino acid structure in your Swiss PDB viewer, and then it will, every time you press control N, it will reduce the energy lower and lower. So once you have get the two, and that will be the layer by layer. Once you have got a layer, that the two consecutive layer, the subsequent energy gap is less than 100 kilocalorie per mole. You have to save that current layer. Okay. Let me check if I have Swiss PDB viewer right now. I might have. SPV. Yeah, Swiss PDB viewer. So once it is open, then what you have to do? You have to open your protein structure and then follow this protocol. It is open now. Now open your PDB file. Our PDB file was saved in desktop demo. Now it is open. Okay. Now what you have to do? You have to select the control. Now this is the layer number 0. It is shown here. Now it is the layer number 0. Now press Control A. Everything is selected. Now press Control N. The one window will appear. It is now minimizing its energy. Okay. It is again calculating the energy for all the atoms. Okay. Then at the end of file, you will find a energy here. So its energy is 16.4 kilocalorie per mole. Okay. Now you again press Control N, and then you see what is the energy of next layer. So now in this case, the energy of this layer is how much? 17.140. So is it under 100 kilocalorie? Is it under 100 kilocalorie range? No. Then again you press Control N. Once the change is not much, then you stop that. Now it is 17,500 and last one was 17,140. So still there is 320 gap, right? Again press Control N. this is the protein preparation. Right now it has 200 gap, 200 energy gap, right? It is 17,737. Our last one was 17,500. Again, press Control N. And then how much you are getting? You are getting 17,913. Again, press. So you have to repeat this step again and again until unless you reach to a state where the energy is not much difference. Right now, the energy difference is less than 100, right? So then what you have to do, you have to save this layer. Okay, save this layer. How to save this layer? Go to the file menu. Go to this file menu. Where it was here in this, right? Go to the file menu. Now save current layer. Where do you see this save current layer? Here, save current layer. Now, if you save this current layer, this will be the energy, minimized energy. So you write EM 4H4F, EM or chymotrypsin. Okay, chyme, and save this in the PDB format. Now your protein is prepared. Okay, now what you have to do? Now you open this. Now you close everything. Now you close everything. You have already saved the layer. Now you have to open your ligand molecule. Now, if you don't have ligand molecule that you can get from the PDB PubChem, so PubChem, you can get any chemical structure here. For the time being, I will only take uh, water or like methyl alcohol, methanol. Okay, because it is the smaller, it has smaller structure, so that is why I am downloading it. Okay, you download the SDF format of it. Download the SDF format, 3D SDF format, and save it in that folder. Once it is downloaded, then what you have to do? Then you perform the docking using Autodock. Open Autodock tools and follow these all steps. What was given here in this protocol. Yeah, docking protocol. So here, once you download the SDF format, then you convert it into mol2 format. Now what you have to do, the docking Autodock is a software in which you have to give the input in their format. Okay, and the format which is acceptable in Autodock is PDBQT. Okay, PDBQT means protein data bank file with charges and with charges and topology, everything. So everything will be saved in that file. So you convert your protein in PDBQT format, your ligand in PDBQT format, then you perform similarly. You save the PDBQT format of your ligand, then you generate a grid box around macromolecule, then set map type. Okay, so Autodock is open. If it is open, then let me see. Yeah, Autodock tool is open. Now, how to load the molecule? You you go to the first file, read molecule. Go to the desktop demo. Your energy minimize structure. So now this is your protein molecule in different format. This is a different representation, which is sticks representation. Now, if you want to visualize in different format, so this will be your ball format. Now this is your surface format. Okay, so likewise you can do. Then you load your ligand, open. Ligand was downloaded in download folder. Oh, that was in SDF format. So that SDF format you have to convert using OpenBevel software. Okay, and then finally, what you do? Then you perform the docking in first you generate the grid, then docking using Autodock. Then finally, when you get the DLG file, then you read that DLG file in the analyze format. Here in docking protocol, open DLG file and then read the macromolecule, which is your protein, and then analyze your results. Okay, so this is what I had to give you a demo about it. Then if you read this document, this is very detailed document, and you will be able to run Autodock by yourself. And if you get any doubt in between, like how to perform these all things, then I will be able to help. You can this is step-by-step protocol for running your docking simulation, and then you can understand what kind of interaction it is a protein is having with the ligand. And okay, so this is what I had to show. Are there any doubts based on today's lecture, or if there are any doubt in the assignment question other than what you have already asked?
Excuse me, sir.
Yes.
In week 6 first video, there was one assignment given by ma'am.
Yes, yes.
She told she will discuss it, but she hasn't in the next video. So can you discuss it a bit?
Yeah, no problem. Yeah, I can do that. What was the question here? I might have this. I think this was the question, right?
Yes, sir.
Okay. Yeah. Let me get a fresh page. Okay, your question was determine the sequence of following pentapeptide. Pentapeptide means it will have five it will have five uh, five amino acids, right? And those amino acids she has given, like the question in the question it is given, it has 2R, then one glycine, then one lysine, and one tryptophan, right? Or not lysine, this is leucine. Okay, so the question now, what this penta then all the steps, all the 4 steps, the digestion step have given some different hints. Okay, so when there was acid hydrolysis of 6 millimolar, what did they observe? It resulted total different uh, five different amino acids: 2 arginine, 1 glycine, one leucine, and 1 tryptophan. So let me first mark 1, 2, 3, 4, and 5. Now in the this has given that these were the in the question it was given the 5. It is a pentapeptide. Now the first experiment has revealed it has arginine, glycine, lysine, leucine, and tryptophan. Now in the second hint, what was given? Reaction of the pentapeptide reaction of the pentapeptide with FD and V followed by acid hydrolysis yielded DNPL. Now, can somebody tell me where the FDNP, which is the degradation, where the FDNP will cleave? This is forming a D, whether it will cleave on the N terminal or C terminal.
N terminal.
N terminal. So it is forming a compound. That means it is in the N terminal, the first amino acid will be L, right? Now you have identified the space of L. So on the N terminal, leftmost side, you will have L. Okay. Now the second hint is digestion of the intact pentapeptide with pepsine yielded free L and a tetrapeptide. Now, what is it suggesting? Free L. Again, it is telling you the L is you had only one L. Okay, and again it is suggesting it is leaving one free L and one tetrapeptide. That means L will be in the left hand side. And where the pepsine digest? Pepsine digest at C terminal, right? What is the digestion site of pepsine? What is the digestion site of pepsine?
C terminal.
C terminal of which amino acid?
Hydrophobic, bulky.
Bulky amino acid, right? C terminal of bulky amino acid. Okay, very good. Now the fourth reaction has given the digestion of intact pentapeptide means without doing any of this, digestion of intact pentapeptide with trypsin yielded free R and a tetrapeptide. What does it suggest? Where will we R? Where the trypsin digest? Trypsin digest at lysine and arginine, right? And after that, there should not be any proline. Okay. So if the digestion with trypsin is releasing one free R, that means R will be on the right hand corner because trypsin digests at the C terminal of a peptide which is having R or what? C terminal of, where does it digest? C terminal of tryptophan, tyrosine, tryptophan, and phenylalanine, right? Bulky group. Where does the trypsin digest?
At lysine and arginine.
Lysine and arginine. Yes. K and R. Not on tryptophan, tyrosine, and that is the nature of chymotrypsin. Right? If I am telling incorrect statement, you should correct me. Okay. So trypsin is digesting at the C terminal of arginine. So it is giving the arginine as a free and one tetrapeptide. That means arginine will be in the right hand side. Okay. Now the third hint, which is the intact and peptide. Now arrange it. If arginine is in the if arginine, one arginine is fixed, another this is fixed. Now where the this will digest? Pepsine will digest after Y, right? Means after L, there should be Y. You have identified the position of Y also. And now what is remaining? G and R. Right. So these two R will be on the rightmost side. Okay. And one G will be here. So your pentapeptide is L, Y, G, R, R. Where the digestion of trypsin will occur? It will occur here. This R terminal. So it will release 1 and 4 pentapeptide, one tetrapeptide. Then if you see this this pepsin will digest after tryptophan, or not tryptophan, tyrosine. So it will give you one lysi, one L and 4 this. Okay. Second statement is also fulfilled. Third statement is also fulfilled. Now your second statement was FDNP followed by acid hydrolysis, which is giving L. So L is on the right hand side. So your correct pentapeptide is L, Y, G, R, R. Is it clear? Is there any doubt? Is it clear to everyone? Pragna?
Yes, sir. It's clear.
Okay. So your correct answer was L, Y, G, R, R. Are there any other doubt related to assignment problem or related to like any lecture material, anything?
Sir, what does pepsin act on?
Pepsin acts on C terminal. Pepsin acts on the C terminal of see, I had given it here. Na? See, the pepsin terminal preference, it is the preferential cleavage at hydrophobic, preferably aromatic amino acid. So where you already know that it is having, this is what I have given here. Na? You already know by your second statement, you already know that L will be in your left hand side. Now, if pepsin is digesting a pentapeptide and releasing one L and another tetrapeptide, that means after L, you will have aromatic amino acid. This is what I had given, right? In the table. In the table, it was given the pepsin preferential cleavage site of pepsin is hydrophobic, preferably aromatic amino acid. Okay, so after L, you will have Y. Is it clear?
Yes, sir.
Okay, are there any more doubt? If not, then we will end this session here. And if there are any other doubt, you can ask that in the WhatsApp group. Okay, I will take this question in the next session, which is on Friday. Okay, Friday 17th of March. This session was conducted in against your when you missed the 10th of March, the live session on 10th of March due to my bad health condition. So now on the 17th, if you have still, if you get any doubts related to your 7th lecture or like previous lectures, then also you can raise that thing in your in the WhatsApp group, or you can email me, and then I will take those doubts in the next session. So if there are no doubts, then I will end this session here, and thank you for joining. I hope you will join in the next session also.
Thank you, sir.
Thank you.